Hello students, welcome back to my class. So here I am back with new video. So dear students, if you remember that in my last class I had discussed you about the Mandel's experiment, right? Incomplete dominance, what is codominance, what are multiple alleles, right? As well as I had sent you some additional videos also where I asked you to watch it properly, where the cell division had been explained, right? So I asked you to go through that video so that you can understand the topic that I'm going to cover today, because here the concept of cell division, the mitotic division, the meiotic division, how the cells divide, the phases of cell division is very much important, because if you are not aware of those concepts, then you will not be able to follow the today's topic. All right, so if you have not watched yet, then do watch the videos that I had shared with you. So with this, let us start our today's class. So dear students, let me give you a quick revision that we had learned. Okay, I had taught you about the Mandel's experiment with the pea plant, which has a different characters with different traits, right? Where he has proposed three laws, right? Law of dominance, law of segregation, and law of independent assortment. From there, we come to know about the incomplete dominance as well, the co-dominance, how how alleles they work, how does they carry the uh, traits from parents to their offspring, means how does it keeps on moving from one generation to the another generation. All right, so in today's class I shall be talking about the chromosomal theory of inheritance. So before we know what exactly this theory is and from where does it came from, who brought up this topic, okay, so for that we should know little history about this. So as I told you that the Mandel's law of inheritance, isn't it? So he published his work on inheritance of character in 1865. So in 1865 he published his work, okay, but what happened, it remained unrecognized, okay, till 1900 it was not recognized by the people, okay. Now what are the reason why was not his work was recognized, okay? So there were several reasons that his work wasn't recognized. So one of them is communication was not easy. So dear students, what happened at that time, see, nowadays we have social media, we have a newspaper, right? The communication is very good at this time, the technologies are very good, but long back these things were not there, okay. So it was very difficult for the people to communicate or to put forward their ideas or their work in front of the people, okay. So one of the reason why his work was not recognized was the communication, okay. So communication was not easy. Second thing was that concept factor, okay. uh Concept of factor, see, he had mentioned, he has explained about the concept of factor. See, if you remember that during his experiment he said that there is a factor which carry the traits, which carry the character from one generation to the another generation, okay, but he could not say that what was that factor, okay. So the factor, okay, his concept of factor as stable and discrete unit that control expression of traits and pair of alleles which does not blend with each other, okay, was not accepted. Why was not this accepted? Because he could not provide any physical evidence. See, what happened, his concept was that there is a factor, okay, and that factor is stable and it is stable and it's a discrete units, and this unit control the expression of trait. So expressing a trait in next generation is controlled by this unit, as well as that the two alleles, okay, see, we know that alleles, they are in pair, right? So you remember this is called your homologous allele, isn't it? And they are in pair, or you can say this is your heterozygous allele, right? Two different allele, and they does not blend. I hope you all remember this independent law of independent assortment, right? So I will discuss with you this again okay. So it is said that the two alleles, they does not blend with each other, and this concept of him, okay, the concept that he put forward, it was not accepted by the other scientists. Why? Because He said it, okay, it was verbal, but he could not provide any physical evidence. See, what happened in science, whenever you discover something new, whenever you invent something new, you have to give a proof, okay, you have to give an evidence that what you are saying is correct, okay. So it is all about the factual. Only saying that okay, this happened because of this is not acceptable in the world of science, okay, you have to prove it. So what happened at that time, the technology was not so advanced, so he could not provide any proof for his concept. Then third was that application of mathematics was totally new and unaccepted by the many scientists of his time. So what happened, he was the first one, right? So for the very first time, he had introduced the maths, the analytics in his experiment, right? But that wasn't accepted by scientists, because what happened, whenever you introduce something new, so it is not easily accepted by the people, it takes time. So this were the reason why the Mendel's work was not recognized till 1900, okay. So after that only his work was recognized. I hope that this is clear, that the reason behind the unrecognition of the Mendel's work, okay, though it was published in 1865. Okay. Then after that, what happens in 1900, okay. So in 1900, what happened, the three scientists, okay, De Vries, Correns and von Sermark. So this were the three scientists. In 1900, what they did, they independently rediscovered Mendel's result on the inheritance of character. See, I had just mentioned the reasons, right, where I had told you that his work was not recognized because the scientists of his times were not accepting his concept, right? So in such case, what happened, the scientists, they rediscovered his concept, okay, they worked independently. Independent means individually, okay, individually they worked, they rediscovered on the same result, on inheritance of character, that whether the Mendel's, okay, whatever he had said is true or not, okay. Then what happened, at the time the technology was advanced, okay, but coming to the 1900, at the time what happened, the technology, it started developing, so advancement came in technology. Then what happened, because of advancement in technology, they were able to carefully observe cell division. So what did they observe? Because of microscope, the advanced technology, they were able to observe the cell division, okay, that how does cell divides. I have shared one video with you, do watch it, okay, there you will know how cell divides if you have not watched yet. So then they come to know that after observation, that this led to the discovery of structure in the nucleus. So when they had observed these things carefully, okay, what happened, this led to the discovery of structure in the nucleus that appeared to double and divide just before each cell division. So what happened, they observed one structure, okay, they discovered a structure, and that structure is discovered where, in the nucleus, inside the nucleus, and that structure appeared to double, okay, it used to get double, replicate, okay, and they divide just before the cell division, means just before the cell divides, that structure also divides, okay. And this structure, so those structure which was observed, which divides before each cell division, they were chromosomes, okay. So now first let us try to understand what is chromosomes, okay, and then we will move to the actual theory of chromosome. See, I'm just going to tell you in brief, okay, what is chromosome. See, this is nucleus. Inside a cell there is nucleus, and inside the nucleus there is the thread-like structure, okay. This thread-like structure is called chromosomes, and this thread-like structure has genes on it, okay. This dot dot dot, I'm just showing it to you to make you understand. So these are gene. So this is your thread-like structure. As you can see, it is all coiled structure, right? This is nucleus and this is gene. And this chromosomes, okay, they carry genetic information. See, gene is a unit of inheritance, right? So when a character is inherited, not only gene transferred, okay, chromosome is transferred. Chromosome goes from parents to the offspring, and along with the chromosome, the gene goes. Means gene and chromosome, they are together, they are not separate. Both of them will move together. together. Okay, this is your chromosome. Now let us try to understand about the chromosomal theory. Now the theory will comes. Okay, the theory of chromosomal inheritance. Okay, so students, I told you that in the year 1900, three scientists, they discovered some structure, right? And they found that that structure get doubled before the cell division, right? And as well, they had learned about the cell division, isn't it? And later, that structure which is present inside the nucleus is called chromosome. And the chromosome, I had just shown you the structure of chromosome, right? It is a trait-like structure, and gene are present in it. So gene and chromosome, they are present together. Okay, so whenever they get transmit, they transmit together. It is not that chromosome will transmit separately and gene will transmit separately. Okay, so they will go together. So from this, what happened then in the year 1902? Okay, so Walter Sutton and Theodore Boveri, so these two were also doing their experiment. Okay, even they were carrying out some experiment independently. So independently, when they were carrying out the experiment, they found the same result on Mendel's inheritance. Okay, the laws of inheritance. So they observed something, and the behavior they observed about the chromosome, that how does it transmit. So they named it as chromosomal theory of inheritance. Now what is chromosomal theory of inheritance? I will explain it to you. So just look at here. Firstly, let us know that what is chromosomal theory of inheritance. So chromosomal theory of inheritance, it allow us to see the relationship between Mendel's laws and chromosome. Okay, the Mendel laws, the law of dominance, law of segregation, law of assortment. Okay, independent assortment. So this things, okay, this three laws and the chromosome, the relationship, that how the laws and chromosome are related to each other, this is what given by this two scientists. Okay, so they studied the chromosomal movement during meiosis. Okay, they had studied the movement of chromosome. Okay, now before I explain you what exactly the chromosomal theory is about, okay, what are the key points of chromosomal theory, let us try to understand this. See, this is a diagram where you can see the germ cell is formed. Okay, now let's get back to independent assortment. Okay, we will do one by one. So first, what happened, the germ cell is forming. Here you can see a blue color chromosome. Okay, and this is the small one, is a black color. As you can see, both are different. See, in your book it is given as your orange, or you can say red and yellow. So I don't have the colored marker, so I'm using blue and black. Okay, and yes, when you are studying this figure, do follow your textbook. Okay, it will be better for you, for your better understanding. So here you can see this is G1 phase. I hope you all know what is G1 phase. If not, then do watch the video that I had shared with you. Okay, and second is your G2 phase. This is your bivalent phase. Okay, why? Because they are, bi means what, two, isn't it? See, here you can say this is a single, right? So here they have become double, two. Okay, they have just doubled. So again, this is called your meiosis 1. Here meiosis 1 takes place, and this is your anaphase stage. All right, this is how the germ cell form. Okay, now this is meiosis 2. If you remember the meiosis 1 and meiosis 2, okay, that the germ cell formed in meiosis 2 stage. Okay, and here you can see this two chromosomes, okay, they get separated. Can you see this? This structure also doubled here. It also become bivalent. And later at the stage in meiosis 2, when they enter the meiosis 2 phase and anaphase, they get separated. Okay, here you can see this gets separated, and then they form 4 germ cell. Correct? I hope that this is clear to you. Now come to the independent assortment. See, in independent assortment, I had taught you one thing, that that gametes, this germ cells, are not dependent on each other. Okay, they are independent of each other. So here you can see, okay, that, okay, let's just start from the beginning. This is blue chromosome, this is black, right? So here you can see that blue and black is going together. Whenever even here they get separated, so blue is paired up with black only, isn't it? It is blue is paired up with black, and black is paired up with blue, right? So it is going in this way. Now you see page number you're in your textbook, page number 82, figure 5.9. Can you see this? Okay. See, as you can see here, okay, at the beginning, you can see this. Can you see this? I'm just explaining it here, okay? I hope you all can see. Okay. Let me just explain it with the help of this. Okay. Okay, I hope you all can see this. Okay. Just look at here. Here you can see this is 2, okay? 2 phases. This is meiosis 1. This is also meiosis 1. This is anaphase. This is also anaphase, okay? Now, these are possibilities. See, here you can see red chromosome is paired up with green, and here yellow chromosome is paired up with red, right? And here also, let us first see about the possibility 1. Here you can see when they get separated, red and green, okay, meiosis 1 in anaphase, they are separated. They become bivalent. Then the germ cell form is red with green and yellow with green. So there you cannot see any change. So they are just going together, right? The way it was paired up here. But you look at the possibility 2. So here you can see that after this stage, when this germ cell are forming, okay, here you see. So you can see the difference. Here you see this is red is going with red, yellow is going with green. So here you see red and green, yellow and red. Okay, red and green, yellow and red. So here you can see yellow, green, isn't it? Could you see this? Yellow, green. So here you see this they are independent. See, it is not necessary that if in a first pair, if red is pairing up with green, in every cases, the red will pair up with green. Here you can see yellow is paired up with green also. Can you see this? It means what? These characters are not dependent. If they were dependent, then in other case also, red would have paired up with the green chromosome. Correct? But here red is paired up with red, and here you can see red is paired up with green and yellow is paired up with red. In second case, you can see that they have just exchanged. Okay, they are exchanged. Could you see this? And this is how the germ cell are formed, that there is a mix and mixture. You see the different pairs, different traits have been coming together. I hope this is clear. Okay, so this is your independent assortment, and this is what about the chromosomal theory of inheritance. So what does the independent assortment tells you about? It tells that when two alleles are together, okay, they doesn't blend with each other, okay, and when they segregate, they are independent of each other. The trait is not dependent on each other. It is not necessary that they will have a same character. Okay, let me give you a day. So now let me give you a day-to-day example of this theory. Okay, based on this theory, you can see among your siblings. Suppose you are 3 siblings. Okay, and out of 3, if you compare, none of you look same. At least you have something different among you, between you. Okay, some might be short, some might be tall, or one of you might be like average height, isn't it? The complexion might be different, the hair can be different. Okay, now what does it mean? It means that the traits that is carrying okay, by the gene or the chromosome, it is that they are not dependent in each other. They can pair up with any other character. Suppose if you say that in your parents, suppose tall and white skin color is dominant. Okay, so if you're thinking that all 3 of you, if you have a white and tall is dominant, that everyone will you have that. Or suppose your mother is short and little brown color. Okay, or it is not that only tall if the height is tall, then the color skin will be white, and it is also not like that the height is short and the color will be brown. It might happen that the progeny may be tall and the skin color may be brown. Okay, or the progeny may be short height with short height and the skin color may be fair, or the progeny may be like of average height and their skin color may be in between. Okay, I hope you could follow me. So this is your chromosomal theory of inheritance. So according to this theory, okay, what does actually this Walter Sutton and Theodore Boveri, what does they proposed about this theory? Okay, let me just read out the point. Number 1, that genes are located at specific location on the chromosome. What did they found? They found that gene has a specific location on the chromosome. See, at the beginning I had told you that gene and chromosome are together, means genes are present on a chromosome, right? So they found it that a gene has a specific location in a chromosome. This is the chromosomal theory of inheritance, first point. Second key point is that chromosome as well as gene both occur in pairs. See, if you remember AA, what is this? Gene means alleles, right? They are present in pair or not? Pair. Here you can see this is a chromosome. Chromosome, they also present in pair. So this is the second thing they found. They had observed in chromosomal theory of inheritance. And then they found that homologous chromosomes separate during meiosis. Okay, here you can see this is homologous chromosome, right? And they are separating during meiosis or not? And they become haploid. Correct? The diploid, then after division they become haploid, right? Then fertilization restores the number of diploid conditions. See, let me tell you one thing about the chromosome, that there are, okay, let me just rub this. I hope that it will be clear to you. There are total 23 pairs of chromosome, okay? 23 pairs. Out of 22 pairs, that is a 44 chromosomes, they are somatic. Okay, they are not reproductive, and one pair, that is two numbers, okay, they are sex cells. Okay, and two numbers means what? This is diploid, right? When division takes place, suppose this, okay, XX, and you know that XY, right? When the meiosis takes place, they separate or not? They get separate, right? The gametes, correct? Then again, what happened when they mate? Means when fertilization takes place, only one gametes will come here. So they become haploid or not? Right? Either this. Only one will come or not. When the fertilization takes place, again they will either become XY or XX. It means they were diploid first. During division, they become half, means reduction division in meiosis takes place. They become haploid, and again after fertilization, they restore the diploid condition. Okay, this is what the chromosomal theory proposed, okay, by the scientist. So, chromosomes segregate as well as assort independently. See, they segregate, right? This chromosome are segregating and they are independent or not. And it is said that they segregate and they independently assort, means they are not dependent on each other. So here as you can see that the gametes are separated, right? And it is not that X is pairing up only with X, isn't it? It is pairing up with Y also, right? I hope you can get it. So they are not dependent on other characters, okay? It could pair up with any of the gametes with any traits, okay? So this is your chromosomal theory of inheritance. So I hope that this is clear to you. What is chromosomal theory of inheritance? So let me give you a brief on this. So what is, who proposed this? It was proposed by Walter and Sutton, okay? Walter, Sutton and Boveri, okay? Just a second. Yes, so Sutton and Boveri, both of them, they were carrying out some experiment, okay, and they found the nature, the behavior of the chromosome. They found that the chromosome behavior is similar with the Mendel's law that he had proposed, okay, and then he found some key points that they are that genes and chromosomes are present in pairs, okay, and they are diploid, okay. During after fertilization, they paired up, they restore the diploid condition, they segregate, chromosome also segregate, but as well as they independently assort also, right? So this is your chromosomal theory of inheritance. So I hope that this is clear to you. So dear students, on chromosomal theory of inheritance, okay, once it is proved that genes are present in chromosomes and the chromosome is the hereditary material and it is carrying the character from parents to the offspring, what happened? So many experimental verification was put forward, okay. So one of the experimental verification was given by Thomas Hunt Morgan and his colleague, okay. So what he did, based on the theory, okay, based on the chromosomal theory of inheritance, they put forward experimental verification. Thomas Hunt Morgan along with his colleagues. So that lead to the word discovering for the variation that sexual reproduction produce. So the experimental verification they gave, okay, from that it was proved that they discovered that variation Okay, the variation that is seen in a progeny is due to what? Variation that sexual reproduction produce. So that is produced by sexual reproduction. And this experiment was done with Drosophila melanogester. So Drosophila melanogester is a fruit fly. So if you have observed, if you keep a fruit, okay, there you will see small flies, right? So that is your fruit flies, and its scientific name is melanogester. Sorry, Drosophila melanogester. So when they experimented on these flies, they found that genes are located in the chromosome. So this was the experiment that was carried out by Thomas Hunt Morgan and his colleague, okay, and this was the proof, experimental proof. So this is all about chromosomal theory of inheritance. So I hope that this concept is clear to you, and even if you have any doubt, then feel free to ask me. Okay, so this is one of the important topic in genetics, so do learn it thoroughly and write notes on this, and if you find any difficulty, then just let me know, and the next topic we will cover in our next class.